So the outline of my talk today is I'm going to be talking about Hadoop architecture and Mapreduce. I'll present particularly a use case on Hive and the protein data bank, some work we've been doing with that. I'll also be discussing another framework which is for parallelizing legacy software for the protein data bank on Hadoop. We'll briefly also mention my future plans with Hadoop, and that involves DNA sequencing and transcriptomics work. And finally, I'll talk to you about a new degree that we've created at Holloway since last year, which is an MSc in data science, and I'm involved in teaching a certain module of that. So we'll cover all of that today. Does anyone have the clicker thing? Because I've got lots of... No,
you want to...
Oh, yes, yeah, please, yeah. Yeah.
It's risky.
Okay, so... Oh, okay, you need to... thank you. Thank you.
I'll need help with that on my time.
Thanks. So is it just forward and back? Oh, brilliant. That's fantastic. I love technology. so big data has resulted in the emergence of a new discipline. And that new discipline is data science. Big data presents huge challenges to computer science, and those are very technical challenges. They're frequently, obviously, summarized by what Doug Laney originally proposed as the 3 V's of big data. I think you've heard them quite frequently. That's volume, velocity, and variety. And some people have even taken that further, and they said that what actually should be, it should actually be described as 7 V's of big data. So variability, veracity, visualization, and value, that extends it to 7 V's. As I said before, my interest in particular is applying big data techniques to scientific data using platforms such as Apache Hadoop, Hive, Pig, and Spark. Now, one of the big problems with scientific data is that now we're in a sort of the era of exponential computing power. You've probably heard of Moore's law that stipulated that every increment of time brings forth a exponential rate of processing power. So we're collecting lots and lots of scientific data, but now we need to process it. We need to process it effectively and efficiently. And with respect to scientific data, one of the key things is to derive scientific conclusions from that data. So you need to be able to have a framework that enables you to do that, to derive robust scientific conclusions that can drive scientific research. So the initial question is what is Hadoop? Essentially, it's a framework that allows distributed parallel processing via the Mapreduce paradigm of large data sets which are reliably stored on a Hadoop distributed file system running on commodity hardware. Now, that's actually quite a loaded description. It's full of information, just that definition alone. It tells you so much about Hadoop, that it's a distributed processing framework. It follows something called Mapreduce, which is it's a new technique of programming to deal with big data. It tells you that it deals with large data sets. There's the word reliably there because Hadoop uses a distributed file system which is replicated across nodes in the cluster, and it can run on commodity hardware. And I guess that's probably very important for you guys because I would highly encourage you to build your own cluster here in Rabat. So what is Mapreduce specifically? Well, Mapreduce is, as I said, a programming model for implementing distributed computing on a massive scale. The essential method is what Frank outlined in his first presentation this morning, which is a modus operandi, so a method of divide and conquer using distributed clusters. So what also is important about Mapreduce is it acts as an abstraction layer. And I heard one person at the front, a business sort of oriented person, talking about how they don't like the nitty-gritty of the programming and managing the clusters and all of this kind of thing. Well, programmers are the same. I mean, they don't want to have to know about which block of data is on which node of the cluster, or any sort of low-level specifics like that, or how communication happens across the nodes of the cluster, or the number of threads or processes running on each machine. So Mapreduce is important in that it acts as an abstraction layer to all of that. It hides the parallelization, the distribution, the fault tolerance. It hides all of that from the programmer. So if somebody's aware of Mapreduce, they don't need to know all the specifics, the nuts and bolts, and the very low-level stuff. And that's one very important thing about Mapreduce. So from this diagram, we see a typical architecture of Mapreduce. So you see on the left side is the data. Let's see if I can find the pointer. So on the left here, you can see the input data. These are workers which are essentially threads that operate across the cluster. So there's the divide and conquer method. And you have the map step and the reduce step, and intermediate files being created in between. So the key features, just to recount, are that it employs Mapreduce, divide and conquer technique, abstracts away a lot of the low-level processes. It's built on a distributed system, and something that Frank mentioned in the morning was the concept of bringing the compute to the data rather than the data to the compute. Because when you bring your data across the network, it creates all sorts of overheads and bandwidth, usage. So the idea is to bring your compute to the data through HDFS, which also makes it massively scalable. One very important thing about Hadoop is that its uptake by technology, industry, and corporations is huge. And there are companies such as Amazon that provide readily implemented Hadoop systems for you to go and use, Hadoop clusters you can use on a cost by use basis. So you just pay as if you're paying for electricity, I think was also mentioned this morning in a previous talk. So that means that anybody can go along and create a Hadoop cluster on Amazon without actually having to touch the physical hardware. And so that's very important because it means that people who have data to process can build a Hadoop cluster without actually touching a single machine, put in their data, and do the work that they want to do. Elastic Mapreduce, again, is another service that Amazon offers that does that. So one of the first areas of science I want to talk about is something called the Protein Data Bank. This is quite a wonderful data set that's accumulated over, I would say, about 20, 30 years. And what the Protein Data Bank is essentially it's models of macromolecular structures. So macromolecular means larger molecules seen in biological systems. So macromolecular structures of proteins, nucleic acids, and complex assemblies. Now, what a complex assembly is when you have say, for instance, a drug molecule binding to a protein. That's what a complex assembly is. All the data in the Protein Data Bank is derived from X-ray crystallographic, nuclear magnetic resonance, and electron microscopy techniques. So high throughput analysis of these structures are a feature of computational biology. Now, this is a very, very important area because computational biology allows scientists to develop drugs to target specific diseases, which you need this sort of resource to be able to tackle these kind of problems and challenges. So that's mentioned on the slides with respect to identifying binders to ligands. Now, this is traditionally achieved with batch-based systems using in-house computational resources. So typically a university will have a cluster that runs very old batch scheduling software that the user, the researcher, the professor or the postdoc submits a query, and it gets put into a batch system for in a queuing system. But now we see new technologies are coming to the fore, which were sort of outlined this morning. Things like Spark, Hadoop, and Hive. So this is actually a typical protein that you see in the protein data bank, or that's actually a cartoon of it. It's actually a lot more complicated than that, but we make it simple by having these cartoonization, so that the spirals are double helices, helices, and the straight chains are called beta-pleated sheets. So the first application I'm going to talk about is a project called Hive Pdb. So Hive Pdb is actually a prototype system for accessing protein structure residues and atomic data through a query language called HiveQL and Python. So essentially, the protein data bank contains about 110,000 structures that have been deposited over 30 years. Now those structures are small files, maybe between a couple of 100 kilobytes and a couple of megabytes. They're text files, and each line of the text file represents an atom in a molecular structure. So a lot of structural biology and computational biology involves querying these positions of atoms and doing calculations on them. So we came up with the idea of using Hive, which is a query language. Does anyone here know what Hive is? Hive? Has anyone used Hive? Okay, that's great. So essentially, Hive is a query language like SQL. It's based on SQL. It's a functional programming language. But the way it's implemented on Hadoop is that when you write a Hive query, it's like writing SQL. So it's quite straightforward to write a Hive query. But then what happens is when you write a Hive query, your query is converted into individual Mapreduce jobs and is run on the cluster in a parallelized fashion. So what we wanted to do was we wanted to provide users, essentially structural biologists, computational biologists, with a method of querying atomic data using Hive query language and also Python. So Hive Pdb was implemented by one of our MSc Data Science students. So our MSc Data Science at Royal Holloway started last year. That was the year that our MSc in Data Science was initiated. And Anna was one of the project students who was assigned to me for the project. I'm in my final year of PhD, but I was co-supervising the student with my PhD supervisor. So, as I said, the aim of Hive was to provide a system where structural biologists can write Hive queries to query molecular data from the protein data bank. And it also supports Python streaming as well. So how is it implemented? So Hive is a system that's often used as an extract, transform, and load system. So that's a sort of concept used in data warehousing. So what we did was we took the protein data bank in XML and we loaded it up into Hive. We chose 10, 100, and 1,000 individual protein data bank entries. So these are molecular structures in 3D format in text files. So we took these files and we loaded them into Hive to test and prototype this framework, because we're interested in testing how fast the system is and how easy it is to use. So Hive was used to write direct queries on the protein data bank data. We employed another method which is called Hadoop Streaming. Now that's very, very useful and important methodology in the specifics of Hadoop, because Hadoop is generally implemented as Mapreduce applications. And Frank this morning was telling of us of his hatred of Java, and Mapreduce programs tend to be written in Java. But Hadoop Streaming allows you to write Mapreduce applications not necessarily in Java. You can write Hadoop applications in Mapreduce applications using Python, Bash, or any scripting language even, using the method of Hadoop Streaming. We actually chose Python, and the reason why we chose Python is we felt that a lot of structural biologists who do computational work like to use Python because it's an easy language to use, and they start to implement solutions in that. So we thought, well, how brilliant would it be if these computational biologists can write a Python program and use a framework like Hadoop to run their calculations on many hundreds of thousands of protein structures at very high speed. So one of the use cases for this framework, so what we wanted to do, we wanted to provide a framework for structural biologists. So essentially a framework that a structural biologist can come along and have a Python program and run it on hundreds of thousands or millions of protein structures in an easy-to-use fashion. So we needed a test case, we needed a use case to test this framework. So one of the use cases we came up with was something called torsional angles, computation of dihedral and torsional angles in proteins. Torsional angles are the angles between the amino acids in a protein structure that give rise to the structure of the protein. So if I perhaps go back a bit to the actual, this diagram here. So you can see from this cartoon structure of a protein that there's various different patterns in the protein. So for instance, these spirals represent molecular structures that are connected together, but they're connected together in such a way that they form a spiral. And the way that they form a spiral is because the angle between this atom and this atom and this atom is of a certain value that puts them into that structure. Now that structure is very important for, knowing that structure is very important for things like drug design, when you want to design a drug that binds to a particular protein. So one of the test applications, if I move forwards now, back to where I was, one of the test cases was computing angles within peptide structures in the protein data bank. So the issues with protein calculations are that it involves measurements between specific atoms in neighbouring residues of molecules. It yields important information about structural conformation that leads to research in drug design. There are 3 such angles that can be measured. And I don't know how many of you have done any chemistry, so there's some chemistry on the bottom left. So this is actually the structure of a dipeptide, so there's 2 amino acids connected together, and on the diagram you can see, I'll use this instead of my finger, you can see that there's phi, psi and omega dihedral angles. So the combination of these angles around these portions of the bond give rise to that structure that we saw previously, you know, the cartoon structure. That all depends on the permutations of these angles. So that was the test case for the Hive project. So what we did was we wrote a program that calculates all of these angles in a protein. So it takes a protein molecule, scans all the atoms, and calculates the number of these atoms. And when we tested it, we found that we compared the Hive system to a traditional batch scheduling cluster. So the traditional batch scheduling cluster that you see in any kind of computational biology laboratory is depicted in red, and the log time is on the left in seconds. And you can see that you can see from the table that that Hive was significantly faster, especially when there are a large number of entries, large number of files. So you can see that essentially that for a small number of files, Hive was slower because it's a system where you need to, there's some overhead in running the program itself. So if you're just going to process a couple of structures, then there's no point in using it. But if you're using a lot of structures and you're calculating, you're performing calculations on a number of jobs, a number of files, then it shows that the that the Hive system is significantly faster than the batch system. So the next project is a very similar project. It's called Pdb Hadoop. Essentially, that's almost the same project, but using Hadoop rather than Hive. We found that the problem with Hive was that SQL doesn't give you much flexibility as to what you can do with the data. So that's why we incorporated Python, so people can write Python scripts and stream in the data. Pdb Hadoop is something I've been working on. So Pdb Hadoop is essentially the same sort of framework that enables structural biologists to run software on all of a fraction of or a fraction of the protein data bank. Now, what makes Pdb Hadoop different is that structural biologists and computational biologists, they work in a very complicated field. The calculations are quite difficult. They tend to be very resistant to change. They use old technologies. They even have programs that are written in Fortran or various other languages. So they have these old programs that do fantastic jobs. They calculate positions of atoms, or they calculate how accessible a region of a protein is for an incoming drug. But these software programs tend to be written in very old languages, and they're not adapted for this new change in technology. So what Pdb Hadoop does is actually the title of this presentation. It allows people to run programs on Hadoop without actually writing any Hadoop code at all. So that's what Pdb Hadoop is aimed at. So why did we do this? Well, the reason was that Hadoop applications must be implemented according to Mapreduce, the Mapreduce paradigm, and this requires specialist knowledge and/or the fact that existing solutions must be rewritten as Mapreduce programs, and that's not something that is trivial. It's actually what computer science calls non-trivial. Pdb Hadoop handles execution of user programs and Mapreduce stuff. So what Pdb Hadoop does is it provides another layer of abstraction to the protein data bank, to the Mapreduce paradigm. So the idea is that we abstract away the need to write Mapreduce. So why is that important? Well, that's important because it means that now structural biologists, people who and computational biologists, people who are doing important work that leads on to drug design and drug development for diseases, can now write programs to run on massive clusters and huge data sets without having to write complicated Mapreduce code, which would terrify them. They just wouldn't. They just wouldn't like to even attempt it. So what we do is we abstract that away through this Pdb Hadoop framework, so that scientists don't have to write Mapreduce programs. They can just plug their program into Hadoop, and our framework takes care of all the complex stuff. So it leverages Apache Hadoop. It's highly scalable, and another feature of the framework is because it uses Hadoop. Hadoop is now being employed by a lot of big companies like Amazon. So as I said earlier in the presentation, the companies like Amazon are providing accounts where you can log in and build a cluster without actually touching a machine. I think Amazon also have free accounts that you can log in, and within a certain amount of data usage you can build a cluster and you can install Pdb Hadoop on it, and you can start analysing biological data sets or the protein data bank. So essentially, to abstract away the Mapreduce stuff, a map step is employed to manage the execution of the user's program. The user just specifies the path to the program, and Hadoop does the job of running the job concurrently. So it massively parallelizes the user's job, as opposed to just doing it batch mode. A user can also pre-process the results from their program. So molecular biology and computational biology programs tend to run on a file and then produce a text report giving sort of information about the structure, maybe binding affinity, how tied to molecule will bind to that protein structure. So it might be a case where a pharmaceutical company has a drug molecule and they want to, or a series of drug molecules, they want to find which one binds tighter to a protein. So they run this program repeatedly on different molecules with the same protein, and they have to sift through the results to get information about which molecule binds more tightly, and that molecule may later become developed as a drug to treat cancer or some medical condition. So Pdb Hadoop allows the user to process the results before the information is stored on HDFS. So one of the challenges with Hadoop is that Hadoop is optimized for large data files. So I think the smallest file size is 64 megabytes, but the protein data bank has very useful information that can range from hundreds of kilobytes to maybe 10, 20, 30 megabytes. So Hadoop isn't really optimized for large numbers of small files, and that was a significant problem in the development of the framework. So we cheated. What we did was we combined all of the small files together into a massive flat file, which was only about 80 gigabytes for 100,000 structures over 30 years of hardcore research. So we used a one-off process to synchronize the protein data bank with a local copy on the cluster. So here we see the architecture of Pdb Hadoop. So on the left side we have the HDFS file system with all the concatenated data, and this shows sort of the Mapreduce methodology of having splits of your data, and then sort of this is where the abstraction happens. This is where all the complex Mapreduce stuff is simplified, so that the user just provides the path to their program, which can be written in any language. It can be written in Python, it can be written in JavaScript, it can be written in Fortran even, although I've not tried that, not tried to run a Fortran program on Hadoop. But the theory is that you could run any kind of old program without having to write Mapreduce. And the post-processing step is a method of maybe doing some kind of grep searching or text filtering on the results. And so how did we apply Pdb Hadoop? So what we did was we tested it. We tested it more vigorously than the Hive PDB project. With Pdb Hadoop, we tested it with 3 types of job. We tested it with a Python script, so that produced the report of these torsional angles. We tested it with a native program that does the same thing as the Python script. So we tested it both with a Python script and a native compiled Linux program. And we also did something quite complicated. We did something called molecular docking. So we used a program called Vena, Autodoc Vena, to dock a given protein structure with the, given drug molecule with the protein data bank, and to provide the scores afterwards. So those were the 3 different types of job that we executed on this framework. And they're the results that we found. We found that Pdb Hadoop, which is in green, is a lot faster and a lot more efficient than just using a regular batch cluster. So the question is, what's next? So my PhD supervisor and I we've been doing a lot of work in DNA and RNA sequencing. In particular, we've been looking at sort of DNA databases. So this is a publication that we've just this publication I've just got published with my supervisor in a top journal, and we want to apply Hadoop to that. So what did we do? So for our research on DNA sequencing, we undertook a detailed literature review of the chemical processes and steps involved in taking DNA from sample and putting it on a computer. So we looked at all the chemical steps that you apply to a DNA sample before it goes onto a computer. And we found that each of those chemical steps can introduce biases in the data. So the three steps that we looked at were fragmentation, ligation, and enrichment. And what we did was we inspected one of the largest DNA databases called the Sequence Read Archive, and we looked at how many of their experiments were annotated. So this Sequence Read Archive, it's several, I think, petabytes or exabytes of data. It's a huge resource. And it consists of 30,000 experiments deposited by researchers in DNA sequencing. What we found that the annotation in this database is poor. We found that out of 30,000 study experiments that were deposited by scientists, only 5.5% contained any detailed metadata about what chemical steps their process used. So what we want to do is we want to apply Hadoop to provide a quality assessment, a QA framework for analysing the actual real data based on the metadata. So we'll look at metadata and we'll look at the type of biases that are introduced in DNA sequencing data, and then we will apply Hadoop to quality assess data in the database. The last slide or two is going to be on teaching big data. So at the beginning I mentioned that Royal Holloway, my university in London, since last year we introduced an MSc in data science, and so these are the modules that students will cover: so data analysis, programming for data analysis, object-oriented programming, database systems, and large-scale data storage and processing. So I actually teach the object-oriented programming and programming for data analysis and large-scale data storage and processing modules on my course. So how do we deliver the sort of disseminating the information and teaching big data to data science students? Well, what we do is we have a number of assignments on a wide variety of areas. So we have Twitter data collection analysis, where you can collect data from Twitter streams. We have an assignment on sentiment analysis using Java and a NoSQL document database called MongoDB. We do some social network graph analysis using SQL, and we get them to do some data mining with Hadoop and Gephi. So we give these sort of assignments to students so that they can develop their skills in a wide range of different areas. My talk was very much on science, and that's why I'd hope that in Chandran's sort of graph of innovation, that maybe my application of big data to science would be a vertical innovation. I would hope, but so then the students perform a dissertation. So that's the content of our MSc in big data. And that's my beautiful university, Royal Holloway University in London. And I think actually on the left that's a picture of our chapel in the university, beautiful university. So thank you very much. Thank you.